I'll begin this afternoon session with a brief introduction. Welcome to the Canadian Institute of Forestry's National Electronic Lecture Program. My name is Sharon Young, and I'll be hosting today's session. Today is Tuesday, March 31st, 2026, and this is the second last session in the e-lecture series entitled Applied Research Perspectives Across the Canadian Forest Service. This series is a collaborative effort coordinated between the Canadian Forest Service and the Canadian Council of Forest Ministers. The CIF-IFC is very pleased to collaborate and host these webinars. For today's session, we are pleased to welcome Funda Ogut, who will present Integrating Genomic Selection with Cutting-Edge Phenotyping Technologies to Support Selection Decisions Under Climate Change. To begin, Funda received her PhD in forest quantitative genetics from North Carolina State University and completed postdoctoral work in maize genetics. Funda joined the Canadian Forest Service in 2020 as a research scientist in quantitative genetics and genomics, focusing on implementing genetic and genomic tools to improve forest resources and support tree breeding programs to support sustainable forest management decisions. With that, I will now pass it over to Funda to begin today's presentation.
Hello. Good morning, good afternoon. Thank you for joining. Like Sharon said, I will be talking about our research as a group, starting with an overview of the multidisciplinary, multi-year, long-term project that we have been working over the last couple of years, mostly focusing on the end-user breeding populations, along with some preliminary results that we got so far. Okay. So we all know that climate change is progressing. It's progressing more quickly than the trees can naturally adapt, and then which is reducing the forest productivity, fiber supply, and as a ecosystem functions. To maintain the healthy and productive forestry under the climate change, it's very essential to deploy the resilient seedlings as part of the climate adaptation strategy. With the frequent droughts, especially very crucial for the first year of the seedlings that deploy in the field, because the water availability is very limited, and then we might end up with loss of mortality. So rapidly identifying, selecting, and then propagating trees with the superior adaptive traits with productivity is very critical for the future forest resilience. Tree improvement programs or breeding programs traditionally focus on the growth traits, but it's not longer sufficient due to the climate change. So we might need to shift the breeding priorities to include adaptive traits, physiological and phenological traits into the breeding programs, along with the growth traits. We all know that breeding programs manage thousands of trees across multiple environments due to the field testing reasoning. But those breeding programs doesn't have enough or capacity for the efficient methods to assess the resilience and to biotic and abiotic stressors. So understanding we have to quantify those traits. The most important role for us, understanding those adaptive traits has any genetical controlling, which is critical for selection and then resilient for the productive seedlings. If there's a genetics, then we can do the selection or make the decisions for the selection for those traits easily. So this figure shows how genetics, genomics are connected in modern tree breeding programs under climate change. It's a multidisciplinary approach, basically. So it's not just quantitative genetics is not sufficient anymore. So we have to combine with the physiology tree physiology with the very novel technologies to be able to collect that data. So genetics focus on the individual genes that are controlling the trait. Genomics is a kind of larger concept, so it's looking at the genes or multiple genes as a whole. So across the genome, examines like the patterns across the genome, which allows us to implement the genomic selection, which means as a kind of genetic prediction. With the genetics, we are more into understand what is the genetic architecture of these traits and then how they are heritable. But with genomics, more focus on the, at least the way we are doing right now, the genetic prediction of the phenotype for future selections. Phenomics is another concept, but it's the study of the phenotypes as a whole under specific genetic and then environmental conditions. So phenomics or phenotype as a result of genetics plus environment. So with the genomics, it tells what potential is there, but phenomics measures or reveals what really had happened. So basically, these are two things because of the environment contribution. Phenomics, again, it's a discipline, but it relies on heavily for the remote sensing tools because we need to collect repeatedly and then rapidly data based on the phenomics. So across many individuals, again, it's not just one or two trees that we are dealing with. Depending on the germplasm, it could be like thousands of trees, unique genotypes, and then everything we are doing at the individual tree level selection. So could be within or among families, depending on the, again, germplasm. But as we know, as a tool, high throughput phenotyping tools can provide large scale phenotyping data. Then we can combine this data with the genomics. And then with this pipeline, if we can develop this pipeline, it will allow us to do some early identification of the genotypes, resilient genotypes, and then accelerates breeding cycles easily. Again, we are trying to combine the genomic selection with the high throughput phenotyping tools. As an, again, multidisciplinary approach, we would like to combine the tree physiology, remote sensing, and genetics genomics together to understand how trees are responding to stress. And then our main focus right now, drought stress, regardless of the species. But we want to start in the control environment and then do the phenotyping and genotyping at the seedling stage, then move whatever we tested in the greenhouse to the field testing for a long-term monitoring for the same traits across different growth stages. Then it will allow us to exclude some of the extreme genotypes, even before moving to the field. In the greenhouse, we have a gantry-based, the same sensor or hyperspectral camera, but it's on a gantry system. And obviously, it's as controlled as possible. But for the field, once we move to the field, it's the real time, whatever happens, depending on the stressors, maybe as a factor of multiple stressors. But we observe those, collect data based on using the drone-based hyperspectral camera. Genomic selection, which is a modern breeding tool or breeding approach that uses the whole genome markers, genome-wide markers, to predict the genetic value of individuals. Again, it's not for understanding or locating the genes that are contributing for the trait, but it's for the prediction as a sum of those effects of the genes. In forestries and in plants, usually we have polygenic traits, meaning that many genes are controlling and contributing to any trait that we are interested in. Accelerates breeding, which can take decades. Improves accuracy because we are using the whole genetic information across the whole genome. Essential for the climate adapted forestry, because once we have the adaptive traits, we can use that data to rapid selection of the resilient seedlings or productive seedlings at the same time with the genomic selection. And the other thing with the traditional pedigree, we cannot capture the within family variation, but because we are using the observed genetic variation using the genome data, we can do the within family selection. High throughput phenotyping tools for the adaptive traits. Photosynthetic and stress-related pigments respond to any temperature, precipitation, or water availability. And those pigments changes under stress, especially under drought. And then they create a unique spectral signatures which can be quantified with the hyperspectral cameras with high resolution. So that's why we would like to use the hyperspectral camera, and then that's the camera we have been using. Again, we are thinking a dual approach. So from greenhouse, we start from greenhouse and then move to the field. Obviously, we are again using the end user breeding populations. They are already established in the field, or we are co-testing with them across maritimes or in Eastern Canada. So, which allow us to do early selection or early detection of the long-term monitoring, and then identification of those traits, adaptive traits, starting from early age, 2 years old we are talking about. And then once we move to the field, that allows us to, depending on the stress that we are testing right now, drought stress trees, how the stress is impacting their growth in the field. So it will allow us to determine that impact. Field testing is a must in a tree improvement program, regardless if you are using genomic data or not. Yes, you can skip some steps, but to keep the prediction model up to date, you have to keep doing your field testing. It's another component of the genomic selection. So whatever we test, we move to field. We are using the Valcartier Research Station, Acadia Research Forest, and then Petawawa Research Forest. And then what you are seeing in here, those sites that we established last spring using the Reciprus families. With the end users, we had a chance for this population. They were already in the field from 2017 test series. So the way we subset it using the same families to be able to make a link from greenhouse to the field, they are already in there. We are working with the Atlantic Tree Improvement Council. The black spruce material that I will be talking about also established last year across 6 sites, 2 in New Brunswick, 2 in Nova Scotia, 1 in PI, and then 1 in Newfoundland. So that will allow us to make a link from the greenhouse material to the field. Obviously, they are putting more families. We can only test subset of those families in the greenhouse. Again, the main goal using the hyperspectral sensor to phenotype the seedlings for the climate extremes, our first stressor is the drought, and then monitor those trees across different growth stages to see if we capture some H-H correlation or quantify the genetic variation in bud phenologic growth and then adaptive traits. But end result would be integrating the genomic selection with the high throughput or rapid phenotyping data to improve the tree selection and then identify climate adaptive genotypes. I would like to give the overview of the current work that we have been doing. Again, since we are working with the breeding improvement programs in the Eastern Canada, red spruce and black spruce are economically important tree species, and then they have tree improvement programs for those 3 species. That's why we tested. Red spruce is a known climate sensitive tree species, and then it's more or less it's a less drought tolerant species compared to the black spruce. We usually, the way we set up on the first year, like the operational perspective, we grow them in their optimum conditions. And then on the second year, we implement the drought stress by decreasing the soil moisture to 40%. So field capacity is usually in the greenhouse at AFC is up to 20% drawdown, but for the drought stress, we selected 40% based on some visual observations as you see in here. Using this, as you see, there is this without creating any cell damage, basically. We collected bud phenology, height data. Drought stress started late June, and then we did bi-weekly imaging. We collected foliage samples for the DNA extraction and eventually for the genotyping, but also we collected every time we image, we collected foliage samples for the pigment quantification. We also collected chlorophyll fluorescence using fluorpen and Lycor 610. We use Lycor 610 for the red spruce and then fluorpen for the black spruce. For the drought stress, we did sub-irrigation for the red spruce, as you see. For the red spruce, we had the Norway's Nova Scotia seed source only. We started on the first year randomized complete block design using 43 full SIP families with single tree plot configuration. So meaning that every cell within a tray represents one family. We started with the 60 progeny across the whole greenhouse. Then after transplanting in 2014, similarly, it was a single tree plot design, but it turned into a kind of split plot in a randomized complete block design structure. So we ended up 24 seedlings per treatment. So 24 for the control, for 24 seedlings for the drought. So in total, 48 seedlings per family. We did the ground measurement, we did the biweekly spectral imaging, and then we genotyped 1,500 seedlings using the 21K SNP chip developed by Fast Track 2, which is co-led by Dr. Lance. He is based in LFC. Some of the families were already in the field, and then Dr. Lance also genotyped as part of the Fast Track 2 project. So we had a chance to look at the genotype data coming from directly field. Yes, there will be age difference, but doesn't matter. And then we already genotype our families. So we will see what is as a kind of beyond proof of concept genomic selection perspective. And then we did the imaging in the field as well using the drone-based hyperspectral camera, foliage collection for the pigment data as well. And then whatever the families we collected foliage, we also did the height measurements. We established our test size last spring. Again, these are the red spruce test trials. Or genetic trials. We would like to assess the adaptive traits across different growth stages. They are already in the field, and then we will see how we are going to monitor them in the long term. For the black spruce, we had 2 seed sources, New Brunswick and Newfoundland. And then in total, we started with 45 full seed families. Again, single tree plot design, randomized complete block design in 2024. After transplanting in 2025, we had 18 blocks per bench. In total, we had the 3 benches. The same 40% drawdown drought stress. Because the end goal moving those seedlings into the field, we didn't want to kill any seedlings at the greenhouse stage since we are genotyping them. We did the same spectral imaging ground measurements, and then this time we genotyped 1,700 seedlings, and then again bi-weekly imaging. The same material will be moved to field this spring, so the seedlings were sent to LFC, and then they will be planted starting in May. I would like to share some of the results. Those are not completed yet because we are missing some pigment data. It hasn't been completed yet. It's taking forever, but I would like to share some results with you now. So for the reciprous heritability, it's the very kind of solid or important concept in quantitative genetics, meaning that what is the proportion of the phenotypic variance that is due to the genetics. So basically that's what it means. So you see the first year and then the second year results in here with the GBLOB. Basically, we using the genomic data, we created a genomic-based relationship matrix, and then we try to estimate, fitting a linear mixed model, their values. So their breeding values, which is a kind of genetic merit of those individuals after partitioning what is genetics, what is environment. Heritability is, again, very important because you can quantify the response to selection and then how much offspring can resemble or will resemble to parents, or that the trait can be evolved quickly under selection. That's how it works in tree improvement. So as you see here, we observed low to moderate heritabilities for different traits, including the germination and then bud phenology. We observed year-to-year variances, changes, or at least not the variances, but changes. And then with the genomic-based breeding values, we were able to quantify some dominance variance, which we cannot quantify based on the traditional pedigree, which is very crucial. We also observed some variation, genetic variation among families. Again, breeding values is the additive genetic value of an individual, the sum of the average alleles, meaning that the genes coming from both of us parents, and then how it carries. So when you do using the breeding value, you will try to understand how much of this phenotype will be transmitted to the offspring, basically. So we observe substantial within family variation for all traits. So obviously within family variation, meaning that individual level differences among siblings. And for these families, there were several families showed higher or lower performances. So there's a way you can do the selection among families as well, not just within. And on the about the HH correlation, obviously from year to year, year 1 to year 2, there's a decrease in terms of the genetic correlation among between ages for the same trait or among traits. So obviously germination or bud set are not really correlated at all, so it doesn't mean anything. And the bud set on the first year and the bud set on the second year is only correlated like 0.57. So it looks like there is a variation and then it's environment dependent, even though there is some genetic control. From the spectral data we collected and then we sent that data. Again, it's not my expertise. We are doing multidisciplinary work. We sent that data to Cornerbrook. We have a colleague, Oliver. He does the processing of that data. We come up with lots of indices, and then you are only seeing in here normalized difference vegetation indices, and then we fit our model, and then looks like it's under genetic control. And then what it means basically it quantifies the plant greenness or chlorophyll content or general photosynthetic capacity or activity. So looks like we captured the genetically meaningful variation in the chlorophyll content, and then as the drought stress progressed, changes in the genetic control observed, but maintained across different data collection dates, even in September as well. So for the chlorophyll fluorescence, to obtain this data, we use LightCore 610, which is a handheld tool to quantify in the light adapted. So it's detecting the short-term plant stress responses, but looks like it has a limited heritability, meaning that not strong candidate for genetic improvement. But again, it might not be a genetically stable trait, but because again, the heritability is kind of close to zero, which is nothing. So is it a strong environmental noise or stress effect? Requires lots of investigation, because again, these tools are not really developed for the short needles. That's what we have been experiencing. And then it's really time consuming. You have to do this data collection within some time frames in the same day, on the same day, and then you cannot extend like height measurements on several days. So lots of factors play a role to collect this data. For the reciprous, treatment effect was significant. So as you see in here, drought stress reduced the growth, leading lower heights, and then causes earlier bud set and then bud development in red spruce. So, which is kind of consistent what the literature says for the conifers in terms of the bud phenology. Yeah. We also, again, I said before, we start to quantify the pigments using the HPLC machine, which is high performance liquid chromatography. So this is supervised by Chris Wang at University of New Brunswick. We have the pigments. Pigments absorb the light and then reflect the light at specific wavelengths, and then that changes occur with the stress. And then depending on the stress, the levels can change. Based on the literature, again, the carotenoid pigments are generally better and then earlier indicator for the drought stress compared to chlorophyll, because it changes occurring in the later stage, but carotenoids respond quickly to any stress, especially for the photoprotection demands. Based on our analysis and the data collection, we only select 2 dates, the first day collection with the image and the last day, to see if we capture something. Looks like from July to September, seedlings show stress induced pigment shifts. And then again, looks like family is very genetically the pigments and stress response under genetic control. But again, we need to finish our other samples, because you are only seeing one rep in here. So to be able to finalize the analysis. The increase in carotenoid pigments suggests the drought stress is playing the role, which we have already observed in the growth data or height measurements. So again, pigments drives the photosynthesis and stress responses, so they are functional traits. And then I think for this study, it's very crucial to understand their role. Once we have the pigment data, we will be linking the spectral indices that observed or collected from the camera with the pigment data, and then we will be able to create a kind of relationship to infer pigment content indirectly from the spectral indices. So again, this part hasn't been done, but obviously carotenoid shows that the families are impacted by the drought stress. For the black spruce, we don't have any genotype data. Actually, we got the genotype data last Friday, so we didn't include that data into our analysis. So we will analyze this data later. But based on the traditional numerator relationship matrix or pedigree-based metrics, we observed low to moderate genetic control again for the height and then bud phenology. We observed the same year-to-year changes, and then we also observed color changes in chlorophyll fluorescence. As drought stress progressed, looks like that like chlorophyll fluorescence under genetic control as well, but we see some variation depending on the timeline. So July is the peak time for the growth, and then we see some clear higher heritability values. For black spruce, seed source effect and then HH correlation, as you see, no significant drought stress effect that we observed with the 40% drawdown. Newfoundland seed sources showed lower growth regardless of the treatment, but statistically it wasn't significant except for the growth in the peak time in the summer, like June, July, it was significant as well. So in terms of the seed source, Newfoundland under drought showed lower growth compared to the New Brunswick control or drought stress seedlings. So for the genetic correlations, HH correlations, the height is still like 0.91. It's not that bad, but we have to keep observing for the upcoming years. For the bud set, it's still under genetic control, but looks like there is this rank variations in terms of from first year to the following year, so year 2, that's why you see 0.73. We looked at the chlorophyll fluorescence distribution under drought stress. Again, we didn't observe anything, but this is the raw data distribution. Again, we didn't fit anything yet. We need to look at other parameters because we collected the data with the floor pan under dark adapted and then light adapted versions. So photosynthetic activity seems like based on the raw data near optimal. Again, we need to calculate the other parameters and then see where we are. We haven't started the pigment data for this project. So genotype data is coming, pigment data is coming. And then for both data sets, we need to do cross-validation to see where we are with the genomic estimated breeding values. Like overall, or to summarize, black spruce somewhat more tolerant based on our breeding populations or our experience with the 40% drawdown. And then red spruce is much more drought sensitive. So selection can be done more on this resilient families. But this multidisciplinary dual greenhouse field approach aims to assess the adaptive traits across different growth stages. Seems like spectral data is promising to get this physiological data or adaptive assessment. And then we need to evaluate the long-term effects of the juvenile drought stress on the conifer seedlings performance. So again, we will start in the greenhouse, end up in the field, and then keep monitoring them in the field. Integrating the genomic selection with the high throughput phenotyping for growth and adaptive traits, we will be able to build a pipeline to process the high throughput sensor and then integrate these high throughput adaptive traits into genomic selection. Once we quantify the genetic variation in those traits, maybe that will improve the prediction and then it will help for the breeding selections. But most importantly, it will enable us to rapid cost-effective selection for resilient and productive genotypes. I would like to thank all the small team, but including the AFC staff, the greenhouse staff, basically John and Greta all the way, because we are mostly working in the greenhouse and then they are helping us a lot. But all the partners and then all the contributors, that's all for me for now. If you have any questions, I will try to answer those.
Thank you, Funda, for the insightful presentation. Well, I don't have a strong background in genetics. I found this topic very interesting. I'll now open the floor for questions from our audience, and I encourage those with technical expertise to share their thoughts. Okay, Fonda, I think we have one question in the chat and I will read it out for you. Hello, Fonda. Thanks a lot for this great presentation. For the field testing, are you using clones for the same seedlings in the greenhouse or different seedlings from the same families?
You hear me? Yeah. So, hello, Omnia. So the thing is, whatever we grow, we are using the same experimental design, and then we are moving them into the field. No clonal material, unfortunately, but if the Atlantic Tree Improvement Council put the same, obviously they are not the same clones, basically, long story short. But since at the family level, we can maybe we will be able to make a link based on their information coming from field or the greenhouse. No clonal material. But the same families, even more families. And then the way we subset those families, we still have a link for the families that we don't have in the greenhouse. So.
Thank you, Fonda. I think we received comment in the Q&A pod, and it was saying it would be interesting to include populations from drier environments such as those further west, for example, northwestern Ontario. What are your thoughts on this, Fonda?
That would be possible, but again, we are working with the end users like Atlantic Tree Improvement Council. So once yes, we are doing this research, but at the end, we are also helping them with our results. They can do their selection decisions based on the breeding values that we are providing them. So if there's a chance, why not? If you can send the seeds, and then if there's a field testing as well, and then once we move those seedlings to Eastern Canada, obviously, are they gonna be alive? That's another question. Or will there be any maladaptation because of the differences like from west to east?
Thank you, Fonda, for answering the question. I think that's all we have so far. If there are no further questions, we'll bring this session to a close. Please feel free to reach out if you have any additional questions for Fonda. Right. Thank you all for joining today's CIF electronic lecture, and a special thank you to Fonda for the great presentation. Bye, everyone.
Thank you.